Hello, everyone, and welcome to today's webinar titled Varicella-Zoster virus, the VZV, one pathogen with multiple clinical manifestations. This webinar is part of the 2021 Microbiology Virtual Week. And I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by DiaSorin Molecular. For more information about our sponsor, please go to molecular.diasorin.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. And if you have any trouble seeing or hearing this presentation, just click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Preeti Pancholi, Director of Clinical Microbiology and Professor of Clinical Pathology at the Ohio State University Wexner Medical Center. For a complete biography of our speakers, please visit the Biography tab at the top right of your screen. Dr. Pancholi, welcome. You may now begin your presentation.
Hello, everybody. It's my pleasure today to give this talk. It's a very interesting topic on herpes viruses and VZV in particular. I am sure we've all encountered herpes viruses in our lifetime, either directly ourselves, with our people that we know, or in our patient population. So it's my pleasure to discuss this very interesting topic with all of you today. So I'd like to start with an outline for the human herpes viruses, and then go straight into the VZV aspects, which is the main gist of my talk today. So 5 facts about human herpes viruses that I'm sure you're all very familiar with. One is that it's a large genome of double-stranded linear DNA that is part and parcel of the herpes viruses. These viruses can establish latent infections and can be extreme viral shedders, as in VZV, HSV, that we're very familiar with. And there are several herpes viruses that are known. However, only 9 routinely infect humans. And these are associated with a wide range of clinical infections that include, dermal, genital, ocular, CNS, organ systems, etc. So wide variety. And then once infected, the human herpes viruses stay latent, which means they pretty much never leave you, and they're part and parcel, I guess they just sort of learn to live with you. So these are the 9 herpes viruses that infect humans, and they're listed here from 1 through 8, and there are 2 HHV6 viruses. And as you can see that close to 100% of adult populations are infected with at least one of these viruses. So you can see how ubiquitous these viruses are. And once infected, one is infected for life. So there are different subfamilies of these viruses. The alpha viruses subfamily that targets the neurons, whereas the beta and the gamma viruses, as shown here, now I don't have a pointer, so you're just going to have to follow along the slide. The beta and the gamma, they target the leukocytes. So there is this propensity of targeting these different anatomic sites. Now, in terms of symptoms and disease conditions, they are very varied. Shown here is herpes HHV 1 and 2, which is herpes simplex virus 1, 2, and VZV. You can see we get cold sores, genital herpes, chickenpox, shingles. And then if you just go down the list of these viruses, you can start to see there are several other manifestations with the remaining human herpes viruses. Now, many times, some of these viruses, they are usually asymptomatic, which means that they do not cause disease right up front. Some do, of course, when you have a cold sore or genital herpes, one is symptomatic. However, as I mentioned, that they do remain latent in the body and can reactivate later in life as a result of an immune compromised condition, old age, or something to that effect that can lead to this reactivation of these viruses. So there's transmission that occurs, primary infection, latency, and then reactivation at some point if these are to be reactivating. Now, it was really interesting to see this article in New York Post about human herpes viruses in the news in association with NASA astronauts. So we know that human herpes viruses have co-evolved with the human being, and they have been a part of our lives for a very, very long time. So it should come as no surprise that this study that was conducted, that was published in Frontiers in Microbiology by this group, they actually studied human herpes viruses in astronauts that were during the shuttle flight, a short flight, 10 to 16 days, or an international space station flight that lasted for greater than 180 days. And they took samples from these astronauts prior to the flight, and then also during the flight, specimens were collected, different body fluids were collected, and then post-flight. And they measured different herpes viruses in these specimens from the astronauts. And what they found was, interestingly, that these viruses were detected, the herpes viruses were detected in these astronauts. So about 53% of the astronauts in the shuttle flights were positive, and 61% of the astronauts from the International Space Station flight were also positive. So these were being shed in their body fluids, and some of these were even cultured on tissue culture, which means that these were live viruses. And interestingly, they also measured stress hormones in these astronauts and shown that they were related to high levels of stress hormones that were also measured at the same time. So, we talk about these being latent and the fact that they can be, expressed during stress as well. So I'm sure my stress levels today, I'm just wondering internally myself if any of these viruses are being activated as a result of this presentation, but maybe not. So just talking about epidemiology of this infection of the herpes viruses, we know it has a very wide distribution. About 95% of adults have been infected with one or the other of the herpes viruses. And just looking at HSV1, VZV, EBV, we know geographic distribution is about 80 to 90%, pretty much all over the world. With CMV, we see a socioeconomic status where 50% we find CMV in 50% of folks in Western Europe and 90% in African and Asian countries. With regards HSV type 2, it ranges from 20 to 70%, and again, based on race and age. So women have more propensity to get HSV2, and also in terms of race, non-Caucasian blacks are more prone to HSV2 or show higher prevalence of HSV2. Now, with regards to transmission of human herpes viruses, we can see this nice pictorial here that talks about transmission occurring as a result of airborne transmission, person-to-person transmission, close contact, vertical from mother to baby, and then also transplanted organs and blood transfusions. So there are just several modalities by which one can acquire these herpes viruses. Now, lab diagnostics is obviously very key and something very dear to my heart since I'm a clinical microbiologist. So we have several different methods of detecting herpes viruses. Viral culture, which used to be something we used to rely on a lot previously. And viral cultures are still very useful, especially if one needs to do antiviral resistance studies. We like to be able to cultivate the virus. But there are other modalities like antigen detection, direct fluorescent antibody staining from a lesion that is infected that can be stained directly. Then there is PCR, which is considered a gold standard for detecting human herpes viruses, especially in the cerebrospinal fluid and CNS infections. And then there is, of course, the histopathological diagnostics, looking for at tissue biopsies and retinal changes as well. Now, with that broad introduction for human herpesvirus, we'll switch now to talking about VZV. Now, VZV, just wanted to just go over a few pointers in terms of just some historical perspectives. Chickenpox was not distinguished from smallpox until the end of 19th century. And in 1875, Steiner demonstrated that chickenpox was caused by an infectious organism because he took some of the material and inoculated volunteers with the vesicular fluid from patients, and they developed acute varicella. So that was interesting. I guess these are things we could, that were done during that era. And then later on in 1888, Von Bouquet, he observed a clinical relationship between varicella and herpes zoster, whereby children that did not have any evidence of varicella immunity, they acquired varicella after contact with a herpes zoster case. And finally, in 1954, VZ was actually isolated from vesicular fluid in tissue culture by Thomas Beller. So you can see it's not so long ago, really, that VZV was actually, cultured in the lab and identified as a causative agent for these infections. So just getting more into the VZV, we know it's a human herpes virus 3. We know it's a very ubiquitous virus because a large number of people are infected with this virus. In fact, greater than 90% of the world population is infected. We talked about it being a double-stranded DNA genome virus. So it targets the T lymphocytes, epithelial cells, and then likes to stay latent in the ganglia, in the sensory ganglion. And transmission occurs through respiratory droplets or direct contact with skin lesions. And interestingly, it has two different forms of disease, one during early childhood or chickenpox, which is generally a self-limiting disease. It's highly contagious and results in itchy, blister-like rash on the skin. And then a secondary infection, which is called shingles, as a result of reactivation of the virus later on in life after having acquired primary infection. And it affects mostly adults, and we have about 1 million cases in the US per year. Now, the virus itself, the VZV virus itself, is a circular virus and has the structure shown on the left-hand side. It comprises of a core region, which is a genetic double-stranded DNA, and it has a capsid, which is a protein coat that surrounds the genetic material. Then that's followed by a tegument, which is the part between the virus and the capsid and the envelope. It's a layer in between. And this contains all the enzymes replicating inside the host cell. And then finally, there is the envelope that you see on the outside with those little spikes on it. And this envelope is made up of modified host cell membrane, and they're viral glycoproteins. And these glycoproteins actually are what helps target it to the neural cells. So they're receptors that bind to these glycoproteins. So just talking a little bit about VZV replication. So you can see here's a virus that is entering the cell, and it attaches to the cell. It gets internalized, and it fuses and uncoats, and then the DNA, it then enters inside the nucleus, whereby it then circularizes and then starts to replicate. And it also produces immediate early proteins or late proteins as a result of this replication. And then it does assemble with the nucleocapsid protein. The DNA then forms these structures, which are then budded outside of the nucleus. They come into the cytoplasm and then go further maturation and assembly to become the fully formed virus particle that is then released to the outside of the cell. So VZV does not integrate into the host chromosome like the retroviruses or adenoviruses. Rather, it's maintained as an extrachromosomal circular episome inside the nucleus. and that's just pretty much how it likes to stay latent inside the cell. So VZV, as we mentioned, is highly infectious virus, and greater than 90% of individuals show serological evidence, which means that they have been infected in the past. And prior to the development of the vaccine, the availability of the vaccine, varicella used to have the rite of passage in children. It meant that a large number of children would get infected, or people would get infected with this virus. And in the early 1990s, there was an average of 4 million people getting the varicella, large numbers being hospitalized, between 10 to 13,000 being hospitalized, and then about 100 to 150 that died each year. After the availability of the varicella vaccine that started in the United States in 1995, there was greater than 90% reduction in morbidity and mortality. So this vaccine is highly efficacious and really helped decrease the disease burden. So talking about vaccination for VZV, this is really a feat and just something that we know is really important in preventing disease. So vaccination was offered in the United States in 1995, as I mentioned. It was offered earlier in Japan and Korea, but we had our doses available in 1995. And there are two different distinct vaccination modalities. One that's given early in life to children. First dose is given at 12 months, and then the second dose is given at 4 to 6 years. And this is with a live attenuated virus vaccine that's produced by Merck. It's the OCA strain. And then this vaccine has also been combined with the measles, mumps, rubella, and varicella as part of that vaccination. So that can be delivered along with as a combined vaccine. So you can see that with the second dose, you have about greater than 98% protection. So it's really highly efficacious. And then there is a second vaccine that is the shingles vaccine that is given to adults. And shingles is given to adults greater than 50 years of age. It's a recombinant non-live vaccine and is given in 2 doses, 2 to 6 months apart. It's also highly effective. It's 97% effective. And more recently, I was very interested to note that it's also recently been approved in July of this year, July 2021, in immune-compromised individuals greater than 18 years. So that is really good news to know that there is something available for a younger age population, immune-compromised situation. Then the other one is the Zostavax virus, which is a live attenuated virus given to individuals greater than 60 years age. It's the same vaccine that's given to children early in life. It's just the dose is much higher. So it's about 14 times higher dose. And it leads to about 50% reduction in shingles, but it's not approved for immune-compromised individuals. So it's just, like I said, a very important step towards eradicating this disease or towards helping against this disease. So now we want to just get into a little bit of the pathogenesis and clinical presentation of VZV. We know it's highly contagious. It's spread by direct contact, or inhalation of droplets of aerosols from vesicular fluids. So here is day 0, where it enters into the respiratory tract and conjunctiva. The virions are then inhaled, and then they enter via this and the mucous membranes. So that's step number 1. After that, there is replication of the virus inside the body, and there is replication that occurs in the nasopharynx and the regional lymph nodes, and then it also reaches the lungs. Now at that stage, now we are at day 4 and 6. Primary viremia results, whereby infected T cells carrying the virus are now able to carry the virus to other parts of the body. So on day 6, 4 to 6, the infected T cells, now move the virus, and there's additional replication and dissemination involving other organ systems like the liver, spleen, the sensory ganglia. So, and then there is a secondary viremia that occurs, which is again part and parcel of the whole virus replication, whereby now the infection can seed the skin. So the whole incubation period and the development of skin lesions, takes about 14 days to 20 days for the skin lesions to appear. So this shows you the varicella, the chickenpox primary infection. So in healthy children, it's generally mild and self-limited. You can see that it starts, the chickenpox starts, maybe in the scalp region and the torso, and then develops on the sides. So it's a really good clinical diagnostics for our physicians to be able to recognize the distribution of this varicella. There's malaise-associated fever associated with this that lasts for 2 to 3 days, and then a large number of lesions, about 200 to 500 lesions, and they can come up in different crops of 2 to 3 crops with 200 to 500 lesions. And these lesions, are in different states of maturation. Generally, some of them are more active and the others crusting, so that you can definitely observe different stages of these lesions. And if someone were to get varicella during adult life, it tends to be more severe as compared to that acquired during childhood. So in terms of incidence of chickenpox, we have about 13 to 16 cases per 1,000 persons per year, and complications about 2 to 6% with acute complications. So worldwide, we have about 140 million cases, and there's about 4.2 million severe complications that occur worldwide, and about 4,000 cases of death associated with varicella chickenpox. So one of the hallmark features of this disease is lifelong latency, as we discussed previously, which is true for all human herpes viruses. So after primary infection occurs, the infection travels from the skin cells, the virions. They travel to the sensory nerve cells. Because of the glycoprotein, that is the mannose-6-phosphate that is located in the viral envelope glycoprotein, it has a corresponding receptor in nerve cells. And so now the virus makes its way into these ganglion, these nerve cells, and then stay latent there and has a propensity to stay latent there for a very long time in the dorsal root ganglion. Now, the virus stays there for a very long time unless and until there is decrease in cell-mediated immunity, either as a result of immune-compromised nature or some sort of immune-compromising situation, or old age, etc. And that's when reactivation can occur from latency, and then the spread of, and then the infection restarts in the form of shingles. So this, I thought, was a really good pictorial that sort of puts it in perspective, the primary infection with varicella on the left-hand side here that we talked about, how the infection occurs, and then the latency of the virus, whereby from the skin, it moves into the neuronal axon, spinal cord, and then lies latent in dorsal root ganglion. And then later in life, it can result into zoster, whereby the virus from the dorsal root ganglion now makes its way back into the skin in the form of dermatomes, as a form of shingles or zoster. So this sort of just really puts it in perspective. Now, talking about shingles, which is reactivation of the herpes viruses, it is generally seen in aging or immune suppression. It's not necessarily true that it's only related to old age or older population, aging population, greater than 60 years old. There is a 8 to 10-fold increase in that population. So there are about 1 million cases a year, as we discussed previously, and that obviously leads to a lot of cost, healthcare costs. Now, in terms of lifelong risk of getting zoster is estimated to be 32%, and 50% of the people living until the age of 85 will likely develop zoster, especially if they're not vaccinated. So I just wanted to reiterate the fact that it's not just only associated with the aging population. I did have a colleague, and we were in our 20s, and she was going to be having her thesis defense coming up, and she was pretty stressed and ended up with having shingles pretty much halfway down her face, which was just very odd, only because we generally tend to think of this as a disease that gets reactivated later in life, but I guess it can happen at any age. So just talking about shingles again, the main symptoms of shingles is obviously the blistery, itchy rash, but you also have, this very painful situation that is called the post-herpetic neuralgia, or PHN, that develops. The rash is there for 2 to 4 weeks, but the PNH, or the post-herpetic neuralgia, can last much longer. So in terms of reactivation with the post-herpetic neuralgia, this just shows you a little pictorial here of how painful and debilitating nerve pain can occur. And that can last actually a few weeks to a year, 2 years actually, and can affect 1 in 5 individuals that have shingles. So it's something really important to keep in mind. Now this graph here on the right shows you the complications with post-herpetic neuralgia that occur as a result of age and sex. So you can see both sexes are affected with the PHN. And as one gets older in life, there is more propensity to have the post-herpetic neuralgia. The incidence is higher. So about in 80 plus years age, the incidence is 33% as compared to 18% if someone is 50 plus years old. So it increases in propensity in an older population. Now, in terms of complications of VZV in general with varicella, which is a primary infection, if varicella occurs in a healthy child, there should be really not too many complications because the immune system, takes care of that. But in rare cases where there is complications, you can see all these different conditions that are listed here, complications with bacterial sepsis, pneumonia, encephalitis, etc., that can occur. With zoster, that is occurring after latency and a reactivation, you can also get many different complications with zoster. And this includes meningoencephalitis, cranial nerve palsies, myelitis, hepatitis, pancreatitis, etc. So these complications can be quite severe as a result of VZV infection. So people that are of a higher risk for severe VZV infections are shown here. So we have solid organ transplant recipients. So it's mainly immune compromised individuals, adults greater than 65 years or older, pregnant women without evidence of immunity to varicella, immune compromised people with leukemia or lymphopenia or on medications, children less than 13 years of age, and also premature babies. So you can see there's a wide list of people with high risk of VZV infections. Now, VZV can also affect, the eyes with Herpes-Zoster ophthalmicus, whereby the optic nerve is impacted. There can be vision loss with that, and then also can lead to permanent blindness in severe situations. We also have the Ramsay-Hunt syndrome, whereby a part of the ear is affected, which can lead to hearing loss, and also muscle paralysis, hearing loss as a Ramsay-Hunt syndrome. So given these things are not very common, however, they certainly can occur. Now, in terms of neurological complications of VZV, this is something of great concern, and I wanted to spend a little bit of time describing these in this slide and the next few slides. So complications, neurological complications occur in 1 to 3 per 10,000 cases. That may not sound like a lot. However, if you consider that greater than population is infected with VZV, that does add up. So in terms of common complications, neurological complications, there is encephalitis, and then they can also be aseptic meningitis, cerebral ataxia, as depicted here on the right-hand side. So several different neurological complications can occur. So what is really interesting that we often discuss is like how a virus that causes no more than a cold sore in one person can actually produce fatal encephalitis in another. So this is just a very interesting question. It's a very interesting immunological, pathological question with a host immune response and the virus interaction. And the same can be posed for COVID infection as to such a diverse manifestation of symptoms. But this is really an enigma and something that a lot of researchers are working on to answer. So in this particular pictorial, I wanted to just show the dorsal root ganglion, which is the latency site, whereby we know that the virus likes to stay latent. Now, in both in immune compromised and immune competent individuals, there can be reactivation at some point in one's life. And depending on the direction that the virus activates, whether it goes towards the spinal cord or the CNS or the brain, or whether it goes centrifugally towards the skin or centripetally towards the brain spinal cord. So it really depends on what path the virus takes that can lead to reactivation of the virus. So this pretty much, I thought this was a nice pictorial that shows you the dorsal root ganglion and then the different pathways that the virus can take. So this is also important to understand that if the reactivation occurs in the spinal cord and the brain cells, it's not always necessary that they have to be manifestations in the skin. So there may not be any rash associated if the virus simply decides to travel into the brain and the spinal cord. So that's why making the CNS diagnostics a little bit more tricky in the absence of rash. So here are some neurological complications of VZV. We talked about viral encephalitis and viral aseptic meningitis. So listed on the left-hand side is viral encephalitis, which is the most common type of encephalitis and caused by viruses. And listed here is VZV that causes 15 to 23% of these viral encephalitis cases, so about 18,000 cases per year, as compared to viral aseptic meningitis on the right-hand side. And you can see VZV along with enteroviruses and other herpes viruses can also cause the viral aseptic meningitis. And there are a total of about 4,600 cases a year in the United States. And this can lead to either, severe complications, permanent brain damage, and encephalitis, or short-term memory loss, etc., during aseptic meningitis. So switching gears a little bit and talking about diagnosis and detection. So diagnosis and detection we talked about in our previous slide, whereby we have culture, we have DFA, direct person antibody, antigen detection, we have PCR, we have histopathological things to do. Now, culture is not so great for VZV because we know that the virus is very labile, doesn't culture very well, and doesn't last outside the host for very long. And also diagnosing breakthrough varicella or diagnosing rashes in general, whether they're breakthrough, whether herpes rash is very hard to do just by looking at the rash. So one of the easiest way to look at diagnosing varicella is by doing PCR of the skin lesions as the preferred test. We know it's easily implemented. We know it's less invasive than drawing blood. And that the specimens need to be collected from lesions. Generally, we want to be able to get specimens early on in infections, collected within 5 days of the rash onset, because that's where you have a large replication of virus occurring. And vesicles are preferred over crusted lesions because, again, the viral loads are higher there. But a good rub on the macule or a papule rash, it could be still sufficient to get an answer. And you generally want to collect about greater or equal to 2 lesions just to be able to get a good specimen. And we know there's really good correlation of sensitivity by doing PCR from the skin lesions. Now, what becomes tricky is when you're trying to do VZV, CNS infections, and you're trying to get, diagnostics for that. Now, we know there are challenges because when somebody has a CNS manifestation, you want to be able to look into, is it infectious or not infectious? With the infectious entities, obviously, we want to be able to find out what they are. Are they bacterial? Are they viral? Now, with the VZV CNS manifestations, we know that these manifestations can occur either as a result of primary infection or reactivation of the VZV. So in both of these entities, CNS can be involved. And then with the CNS complications, we know that these complications can occur without having a detectable lesion or a zoster rash in individuals. So that makes it a little bit difficult clinically to diagnose these infections without having good diagnostics on hand. So thereby, rapid diagnostics for these type of manifestations are critically important. So looking at the encephalitis diagnostic guidelines that are out there for all of us to review, it's along with HSV, you can see that VZV is also indicated as a virus that should be looked for in these encephalitis cases. And PCR is preferred, is a preferred test to do for these virus infections, HSV, VZV as well. So it is recommended that a CSF sample be obtained, and then these PCR assays are highly sensitive and specific, and they have a very fast turnaround time and require only a minimal amount of CSF specimen, and should be carried out as soon as possible. And in the absence of a PCR being positive, one can also look for VZV-specific antibodies in the CSF that can be utilized as well. Likewise, for meningitis, we want to be able to distinguish viral meningitis caused by HSV, VZV, enterovirus, arboviruses from those caused by bacterial agents. It's really important to differentiate these because, again, that will reduce the use of antibiotics, it decreases the length of stay, prevents further spread of the infection, getting these diagnostics squared away. And within the viral meningitis panel, as you can see, along with herpes and enteroviruses, there's also VZV that should be looked at, again, by PCR or by DFA or by Zank smear. But obviously, PCR of the CSF is the preferred approach. Now, in terms of the different assays that are available for VZV infection, there are several that are available commercially. So I've listed the ones that are out there. One of which is the BioFire, which is the meningitis encephalitis panel. It contains 14 bacterial, viral, yeast pathogens all combined in a multiplex format, and the sample here is CSF. And then we have Quidel assays, the Lyra, HSV 1 and 2, and VZV combined, and the Solana assays. And here they're looking for HSV and VZV together, and the samples is cutaneous and mucocutaneous swab specimens. So as you can see, sometimes it's hard to differentiate, HSV from VZV, hence the need for having a combined assay that can look for both of these entities. And then there's the DSRN molecular assay, Simplexa assay, which is the VZV direct assay for CSF. And there's a VZV swab direct assay that does cutaneous and mucocutaneous swabs. So that covers a wide range of different sample types. And they're all FDA approved, FDA cleared. So in the form of a summary, we know that VZV causes varicella, chickenpox, and also Herpes-Zoster, which is shingles. VZV is highly contagious and can be spread to others very easily since the virus is shed from the varicella or from the zoster infections. It can be spread by person-to-person contact or by inhalation of aerosols from vesicular fluids from the skin lesions and also through respiratory secretions. So several different ways. And so this definitely requires isolation in the hospital if somebody is suspected of VZV. VZV, central nervous system, can have various manifestations, various presentations, including encephalitis, meningitis, and identifying VZV becomes critically important in these individuals to prevent morbidity and mortality, and especially more challenging in these patients if they present without a rash. We talked about VZV CSF PCR as being playing a pivotal role in making rapid and accurate diagnostics, which are very essential for treatment of VZV. And then finally, we talked about also the VZV vaccines as being very effective in preventing disease sequela for both the primary chickenpox and also shingles disease. So with this, I will stop and take any questions. Thank you for your attention.